in regenerative medicine, we want to generate something new. Okay, so it could be a cell, it could be like a couple of cells, it could be tissues, organs, of course, and specifically, regenerative medicine of the kidney is trying to generate new cells, new parts, the entire organ, which is the kidney, of course. So it's focused on the kidney. So you want to generate something new. So how do you generate something new? So there are basically two options. You need to learn first, basically, how the kidney intrinsically generates new cells or replenishes its own compartments. So if you understand that, okay, what we call the regenerative programs, the intrinsic regenerative programs, you can try to introduce molecules, therapeutics that would, I would say, activate these intrinsic regenerative or repair programs that eventually will generate new cells in the kidney. So that's one way to skin the cat. The second way is to not to cultivate on intrinsic regenerative programs, but rather introduce something from the outside, build a kidney outside, build a kidney part outside, or just, inject cells, what we call cell therapy, into the kidney that would generate new kidney cells. So we call that, for instance, exo-regeneration or intro-regeneration. Exo being delivering things from the outside that we generate in the lab, or intro is something that you a way to cash on the intrinsic regenerative programs in the kidney. So there, there's breakthroughs all the time in that regard. I mean, when you think about the regenerative programs, there are it's more a basic science showing, again, the mechanisms of kidney regeneration, specific molecules, specific repair programs. And once you understand these pathways that are activated in these, I would say, in these when complete regeneration occurs and new cells are appearing in the kidney, you can, once you understand the molecular biology of that and the cell biology of that, you can use ways to activate it. So, there's just been a big publication about, what is a faulty regenerative program? A program that leads not to generation of new kidney cells, but rather generation or induction of fibroblasts. That's not good because you don't want to have chronic kidney disease, as opposed to a program that is full, complete regenerative program that ends up in regenerating new cells in the kidney. So that there's just a paper out about the differences between a faulty regenerative program leading to maladaptive and faulty repair leading to chronic kidney disease, as opposed to regeneration, getting new cells and fixing the kidney up by intrinsic mechanisms. That, so there are other, additionally, more papers showing, again, different approaches of cell therapies, trying to get stem cells, kidney stem cells, or non-kidney stem cells in the kidney, and to generate maybe new cells, or even just the cells would secrete factors that would induce repair. Or there are attempts at bioengineering whole kidneys outside our body, or kidney parts in that matter, again, with tissue engineering techniques. Well, you build, you mix up kidney cells with extracellular matrix outside the body, and then you get these kidneys that are then, these are kidneys that are built in the petri dish, and then they're being grafted in our own kidneys. So the field is flourishing, and different, I would say, ideas are being, brought about and are advanced, and we're waiting for clinical successes also. Okay, because it's still not in the clinic, and I'm a kidney doctor in addition to being a stem cell biologist, so I want my patients to enjoy, the field of kidney regenerative medicine. Well, we have many projects that are related to kidney regeneration. Specifically, we've been for many years interested in how the adult kidney is replenishing itself. And for instance, we discovered that each compartment in the adult kidney is responsible for replenishment of by specifics, by specific cells that act as progenitor cells, and they it's like these, it's called local regeneration. It's not building, during fetal life you build whole nephrons, but in adult life you just regenerate by yourself different compartments, proximal tubules, distal tubules, collecting ducts, loop of Henle, by cells that can go into the cell cycle and self-renew and introduce new cells. So interestingly, some of these cells, I would say, they're not born as kidney progenitors, but they can be activated to be a kidney progenitor. They can be found in the urine. Okay, so interestingly, we were very interested in collecting urine from patients and deriving kidney progenitors from their urine. And when you have kidney progenitors in the urine, which are shedded from the kidney to the urine, you can actually expand these cells, get millions of cells in culture, and use them to build what we call organospheres, or these, we culture them as three-dimensional structures, and then you can make them into spheroids. This is more of a cell aggregation, or you can make them as organoids, as kidney organoids, and it's really neat because then you have from your own urine a representation of your own kidney in the petri dish, and that's amazing because you can, when you have this representation in the petri dish, it's your own urine. It's non-invasive, and we work very hard to get these cultures up and running and understand what is the methodologies to grow these 3D kidney organospheres from the urine. Once you have them, you can learn about your disease, okay, in vitro, but you can also use these mini kidneys for regeneration. So once we establish the protocols for growing urine-derived kidney organospheres from one's own urine, we started learning about the disease of the patient, whether it be an acquired disease or inherited disease, a genetic disease, or we use these mini kidneys in a dish for as grafts for kidney regeneration. And it's quite remarkable how these small kidneys are so informative in terms of disease modeling and kidney regeneration. And it's just urine, we don't do a biopsy, we use urine. The regenerative medicine field is sort of has to move right from preclinical studies and from early clinical studies to more, to more advanced clinical phases and get approvals. Yeah. So remember, things take time. CAR T technology, for instance, which is implemented now in cancer and autoimmune disease, the first discoveries were in the 80s of these, how to generate CAR Ts, So there is this lag time between, right, science and medicine, and we have to do things carefully and in the right way. So I think that in a matter of years, there will be the regenerative medicine field, and specifically within the kidney, will flourish. It will take, there's this point where the point of no return, where things will be, the basic and translation science would be, at a point that would allow more and more clinical studies. But for instance, we're understanding that these mini kidneys that we generate secrete repair factors. Okay, we generate these. We take one's own urine or one's own kidney cells, and we generate these mini kidneys, and they're very interesting. They're very interesting for kidney regeneration when you implement them as grafts, but they are also secreting repair factors. These mini kidneys are secreting repair factors. Now, these repair factors are crucial both for prevention of fibrosis and activation of these regenerative programs that I discussed early in our talk. So it's very interesting. I mean, it's not cell therapy, but it's the mini kidneys we grow outside of the body to understand what are the molecules they secrete that allow kidney repair and kidney regeneration. And this is once, and we understand more and more what are the secreted factors that do the job, because again, these kidney progenitors generate many kidneys, but I don't graft the many kidneys, but I rather study what the kidney organoids are secreting. And these factors, again, are repair factors, regenerative factors, and they can be moved onto clinical studies very rapidly. So in a way, you can also use these cell-based assays that we study in the lab to really derive therapeutics that which are not cells themselves, but rather secreted factors by cells that will do the job. And this is something that could move very forward very fastly into the clinic.